So first, I want to thank Andreas for the invitation, but also in particular Julie Coughlin and Dave Hanlon at Quanterix. It's through a program that we have with Dave's accelerator program that we have been able to, what I would say, accelerate some of the fluid biomarker discovery that we have done at Biogen. So before I talk about some of the data associated with that work, I'm just going to introduce some of our focus that we have ongoing at Biogen, as well as our definitions and where we're trying to spend a lot of our focus in developing novel biomarkers. I did want to just comment that at the moment, Biogen is committed to studying neurology and developing novel therapeutics in the space of neurological disease. In particular, we are targeting Alzheimer's disease, ALS, as well as Parkinson's disease. And in particular, in the space of Alzheimer's disease, amyloid and tau are our two main targets, as is very common in the neurodegenerative AD space, as well as in ALS, both a concerted assault on familial and sporadic ALS. And then finally, trying to develop new targets, both biomarker endpoints as well as therapeutic targets in all three of these neurodegenerative diseases. So before I jump into some additional background on our biomarker classification, I just wanted to emphasize that at Biogen, we are actually committed to biomarker discovery. What we're trying to do here is invest in biomarker discovery and development as early as you possibly can to partner and pair with our programs as they progress through the pipeline to be as successful as possible. And one of the things that we've observed and I've identified over time is that this innovation is inversely proportional to validation. Oftentimes, validation can get in the way of developing novel biomarkers and can get in the way of innovation. Novel targets rarely have validated biomarkers, qualified or validated, and oftentimes when you're developing those novel targets, you should be developing the biomarker activities alongside of those novel targets. We have a critical investment in developing these biomarkers alongside the development plan. So it's not just about the safety and tolerability that you might see in a phase 1, it's also that critical investment in the biomarker endpoint. So as we heard both in the neurology panel from this morning, but also from Jessica, it's important that you define the biomarker that you're talking about. The term biomarker is used interchangeably in a number of different ways, but biomarkers are used for a variety of different purposes. So before I go through some of the data that we've generated with Quanterix, I want to define Biogen's classification of biomarkers. We bucket biomarkers into three main categories: pathway biomarkers, disease biomarkers, and safety biomarkers. So with respect to pathway biomarkers, we're specifically talking about biomarkers of target engagement or a pharmacodynamic response. So this could be in a healthy volunteer population or in a diseased population. This biomarker tells us that our drug of interest is getting to the target region and having the biological impact that we would expect. Disease biomarkers can be used either for patient enrichment, patient selection, and or to track with disease progression over time. And then finally, safety biomarkers. Not just are we interested in identifying the right patient population and the right treatment response, we also want to be able to use biomarkers that will help us to identify when an individual might display a safety event. And I think it's important that although the talk today will focus on the work that we've done in the fluid space, we are agnostic to the modality. I mean, we are interested in fluid-based measures to identify novel biomarkers, preclinical and clinical imaging to identify novel biomarkers, as well as human physiological devices. So in the case of Parkinson's disease, wearables would be one example. Just a couple schematics before I get into some of the data. We do feel strongly that biomarkers will enable early proof of biology. And so as you can see depicted here, whether that's nerve muscle physiology, visual evoked potentials, EEG, imaging, whether tau PET, amyloid PET, fMRI, or even CSF fluid endpoints such as A-beta, tau, alpha-synuclein, and some novel ones that we'll talk about today. The questions that we're hoping to answer by engaging in this early proof of biology, as I've mentioned previously, did the drug engage the target? Have we selected the right dose? So not only are we looking for confirmation that our drug has impacted the target, we want to know if we can use that biomarker then to identify doses for our subsequent trials or even second cohorts that might give us a greater or changed response. And finally, did the drug impact the biology of interest? In addition, not only are we interested in the good questions I just mentioned, we're also interested in identifying the right subpopulation of patients. From our current aducanumab drug in Alzheimer's disease, we've identified that there's a particular particular population of patients that are going to respond to that therapy. Using that patient selection criteria, we've had success in our current early clinical trials. Can the biomarkers that we've measured provide insight into disease mechanism? One biomarker might be useful as a patient selection tool, but also might be useful as a disease progression biomarker, as is the case with tau. It looks like in the Alzheimer's disease space, tau accumulates over time. Whether it's through a spreading hypothesis or some other biological mechanism, the increase in tau does seem to increase as the disease progresses. And finally, can biomarkers provide support for efficacy? So as I mentioned at the beginning, through a collaboration with Dave Hanlon and Julie Coughlin at Quanterix, we have been working with their accelerator program to what I would call accelerate our early efforts in the fluid neurodegenerative disease biomarker space. What I'm going to talk to you about this afternoon is specific work associated with developing what I'm calling the pathway biomarkers, so specifically those target engagement pharmacodynamic endpoints. And because we are focused in the neurospace, I'm specifically focused for the rest of the talk will be on CSF as our matrix of interest. It doesn't in any way, as you heard this morning, and also from Jessica's talk, we are also actively looking for blood-based biomarkers of disease, but when we're talking about our pathway biomarkers or our pharmacodynamic endpoints, we really want to see that we can have that change in a matrix that's closer to the CNS, so we're looking in cerebrospinal fluid. So as I've mentioned, we've been working with Quanterix utilizing their Simoa technology, and so I'm going to share with you three examples of the collaboration that we've had with Quanterix over the last 12 months or so. The first one is in the tau space. Unfortunately, there's this odd little box in the middle of all of my slides that seems to be blocking some of the text. I apologize. So before I jump into what we've done, I just wanted to, I think most of you are familiar with the tau space, and in particular the existing tau ELISAs that are out there for the Alzheimer's disease therapeutic area. But I did want to highlight those because it is relevant. The existing assay that most people use to measure tau in CSF or in blood are either the in-a-test ELISA, which is a measure of total tau using the HT7 and the BT2 antibodies, or for phospho-tau, it's HT7 and AT270. And then for the newer assays on the Simoa that allow for the measurement of tau in blood, primarily in plasma, there's the Quanterix 2.0 assay that's on the Simoa platform. So we were actually interested in developing a new tau assay using novel Biogen antibodies to measure tau in cerebrospinal fluid. And the objective really was to use this assay as a pharmacodynamic endpoint in our clinical trial to enable go, no-go decisions after phase 1. So that first program that I'm going to describe for you is an antibody that we have, which is listed here as BioAB1. So it's just Biogen antibody 1, and it's a pan-tau antibody. So through the Quanterix Accelerator program, we provided Dave with this antibody, as well as talked through with his scientists the number of the other tau antibodies that are in this space today. And the Quanterix Accelerator program paired BioAB1 with HT7, BT2, ATX, a number of other pan-tau antibodies. Also, in addition to that, they used the tau calibrator that comes in the 2.0 kit, and they developed the optimal assay parameters, both the incubation time, the number of washes, coupling of the beads, etc., to come up with these four different pairs that you see here. The first pair is the ADX202 antibody and our antibody as detection. The last three pairs are using our antibody as capture paired with various other pan-tau antibodies. We did a number of other experiments to, if you can see looking at the limit of detection here, they're all fairly comparable, very low picogram per mil. We did a number of other experiments to identify which one to move forward with, but all four of these looked fairly comparable. So we selected the first pair, the ADX202 as the capture, and the Biogen antibody as detection, and followed that with some ex vivo experiments. So in this set of experiments, what we did with that antibody was we took healthy control CSF as well as AD-CSF. And with those CSF samples, we spiked in different concentrations of our drug. We expected that in the presence of that drug, we would see a reduction in the levels of tau in a concentration-dependent manner. So if you look at the y-axis, you can see the levels of tau measured both in AD CSF and healthy control CSF. And the levels of tau, if you look at just in the absence of any drug, are comparable to what you would expect if you were to use the INA-test ELISA, slightly higher in the AD population. And then if you look at the y-axis, with different concentrations of drug spiked into CSF, you can see a reduction in the level of tau in a concentration-dependent manner. The important thing about this study for us was that for our clinical program, we wanted to ensure that the assay that we were using to enable a go, no-go decision would not be affected whether or not we used AD-CSF, which can have higher levels of tau, versus healthy control. And while the y-axis is both 200 picograms per mil, you can see the same level of percent reduction, which equates to 99% at the highest concentrations of our drug. So this experiment was followed by an in vivo experiment in non-human primates, which would allow us to see the sort of percent reduction following a systemic injection of our drug. So quickly, the methodology that we used, CSF was collected from non-human primates at either 8 days before injection or 2 days before injection. So we took 2 baseline measurements. They received an injection of our drug or vehicle at time 0, and then repeatedly up to 1,000 hours, which I think is roughly 6 weeks later, maybe longer than that, we collected CSF to look for a change in tau levels. The data that we generated is shown here on the bottom left. Tau, again, using the assay that we developed with the accelerator program, is on the y-axis, and the time from post-dose is on the x-axis. So first, I just want to draw your attention. Each individual circle represents an individual animal. So the baseline levels of tau in these cynos is roughly 50 picograms per mil. At 2 hours post-injection, the levels of tau are comparable to what they're seen at baseline. However, at 24 hours and persisting through 168 hours and starting to return to normal at 248 hours is a reduction in tau. So using this assay, we were able to confirm in CSF that administration of our drug would reduce the levels of tau, providing us with a measure of target engagement and pharmacodynamic response. So the second example from the tau space that I'll provide is not as straightforward, and unfortunately we've run into a number of challenges. The second activity, we needed to develop a novel assay to measure phospho-tau. The existing phospho-tau assay that's in the literature and in the commercial space is against tau 181. We needed to develop an assay for a different epitope than what's published. So working again with the accelerator program, we provided them with an antibody. In this case, it's Biogen antibody 2. They did very similar activities to what they did for the pan-tau assay. They paired Biogen antibody 2 with a number of pan-tau antibodies. Unfortunately, as is the case when you're developing a phospho assay, there's no good calibrator or standard available to use in assay development. We did explore a number of different activities. We took recombinant tau and incubated it with known kinases that are thought to phosphorylate tau at this particular epitope. Unfortunately, the levels of tau phosphorylated at that epitope were too low to use as a calibrator. We developed peptides with New England peptide that are supposed to bind both the epitope of interest for us as well as the tau antibodies that are pan-tau antibodies. But unfortunately, to date, none of those activities have been successful. So we do not yet have a calibrator that we were able to use in this assay development. So instead of using a recombinant protein or a peptide, we decided that we would, so that we didn't delay assay development, we would use transgenic mouse brain. There are a number of tau transgenic mouse models available, and most of those tau transgenic mouse models express the phosphorylated tau of interest at very high levels. So we used brain lysate from the tau transgenic mice as our endogenous control, as well as different concentrations of that to come up with a calibrator. So through Quanteric's accelerator program, we screened multiple antibody configurations against the phosphopeptides and the transgenic brain. We selected the best performing pair based on that data with the endogenous positive control, and then we moved forward and we tested both healthy volunteer and AD-CSF. So before I show you with the CSF data, I wanted to just highlight again this approach to developing this assay in the absence of a calibrator or the absence of a standard. So as I mentioned, we used brain homogenate from tau transgenic animals. And the brain homogenate is depicted here in this Western blot. So animal number 1 has phospho-tau levels that are elevated. Animal number 2 also has phospho-tau elevated levels that are elevated, but not quite as much. So we used the brain homogenate from these animals and then determined the total concentration of tau as well as the total concentration of phospho-tau in these brain homogenates. From those brain homogenates, we developed a standard curve based on the total phospho-tau 181 levels. And so you can see the curve here as generated. So from this point, again, in the absence of a better calibrator, we used the brain homogenate to move forward to see if we could determine any signal that was BLQ or above background relative to the brain homogenate as our calibrator. So next we tested AD-CSF. Both AD-CSF as well as healthy control CSF. We did 2 different sample sets. The graph on the left, you can see that in the first data set that we saw, it looked like there was a slight elevation in terms of AD-CSF levels of this particular phospho-tau epitope. And in fact, it looked like there was a multimodal distribution relative to healthy controls. Of course, we wanted to repeat that and see if that phenomenon was in any way believable. And we ran a second set of CSF, and there was absolutely no difference between healthies and AD. And I also have to point out that these ADs and healthy CSF samples are the red dots here on our calibration curve. Again, it's a calibration curve using brain homogenate, so it is suboptimal. But regardless of that, you can see that these levels are all BLQ. They're all basically at the limit of detection. So they're very low levels, basically at background. And because of the lack of the calibrator or the standard, this project is especially challenging. At the moment with Quanterix, we have halted efforts until we can come up with a better calibrator, and we have a number of activities ongoing to do so. But even in the presence of a better calibrator, it's hard to determine whether or not we can even push that sensitivity level any lower to be able to see a meaningful measure of tau in AD and healthy controls. So the final example I will describe today is a program associated with a novel biomarker of interest to us, and that's targeting ataxin-2. Ataxin-2 is a protein that's involved in spinocerebellar ataxia. Mutations in ataxin-2 can cause spinocerebellar ataxia. So in this particular program, it's very similar to what we've done before. We provided Quanterix with 3 commercially available ataxin-2 antibodies. We provided them also with partially recombinant protein as well as an ataxin-2 fusion protein. And in this particular case, because we had no evidence that this protein of interest would be present in CSF, we also provided them with positive and negative cell lysates as controls, both positive and negative cell lysates as well as knockout tissue. So the first task was to have the accelerator program screen the number of the different antibodies that we provided using our positive and negative controls as well as our recombinant proteins. After that, they selected the best performing pair and then continued with the assay development and optimization. And then finally, we provided CSF samples from a particular neurodegenerative disease as well as healthy volunteers to see if we were able to measure the levels of this protein in CSF. So in terms of the assay development, when we first started these activities and provided the cell lysates, we provided both positive cell lysates as well as a negative control, which is a knockout tissue. So if you look at the graphs, these 3 graphs are basically representing the same cell lysates. The red dots are knockdown tissue, and the blue dots are positive tissue lysate. The 3 different panels are the 3 different antibody pairs that we explored. In the case of the top right, it's antibody 2 as the as the capture, as well as the detection antibody. In the bottom left, it's antibody 2 as the capture and antibody 1 as the detection, and then a third pair, 2 as the capture and 3 as the detection. So the first thing that you might note from looking at this graph is that we were expecting to see a reduced signal in the knockout. The 2 graphs on the bottom left don't show any sort of reduction in the knockout tissue. If in fact, the levels in the positive lysate are actually below the levels in the knockout lysate. So the only antibody pair, and there's another point that's off the curve here, the only antibody pair that actually showed a reduction in terms of the levels in the knockout tissue was antibody 2 as capture and antibody 2 as detection. So we moved forward with this antibody pair. In addition to doing some other optimization, we also explored another calibrator. In this particular case, it was an expanded fusion protein with a GST tag, and as a result, we were able to expand our curve and produce a limit of detection for this particular analyte at 0.65 picograms per mil. You can see the absorbances over the concentration of the standard here on the right. And then finally, using this new assay, we tested some neurodegenerative disease CSF as well as normal control CSF, and we're able to confirm the presence of ataxin-2 in CSF samples with a slight shift in, or slight reduction in the neurodegenerative disease of interest. So the results from this experiment demonstrate that in our 20 CSF samples, we are able to detect ataxin-2, and the assay optimization for this particular assay is ongoing and will be used in our clinical program going forward. And I think to summarize, I just wanted to, again, share with you 3 different examples of assay development activities that we have undertaken with Quanterix and the Quanterix Accelerator Program to what we're calling Accelerate, our fluid assay development activities at Biogen. I think we have a couple examples of some successful activities, as well as an example of what I would call a more challenging activity in the phospho-tau space. It's possible that this is challenging not because we don't have the right reagents, but because the levels of this particular analyte are just not present in CSF. And we will continue to work with the Accelerator Program going forward for additional activities. And then finally, I did want to thank Dave Hanlon and the other members of his team at Quanterix, as well as the team at Biogen for the data that was generated and shared with you today.
Do you have any estimate of how much better the sensitivity is regular to regular ELISA?
How much improvement of sensitivity relative to what? I'm sorry? So for tau, or so in terms of just the assay developments that we've done, we have not done a direct comparison of, let's say, our pan-tau assay relative to the INATEST assay. However, if you just compare the work that Quanterix has done in terms of the improved sensitivity, it's on the order of 300 to 1,000-fold improvements. But we have not done that comparison for these novel assays. So are you specifically asking about pre-analytical variables that might impact the quantification of these assays during the proteins that aggregate? So this is a major factor for something like amyloid at A-beta 42. For tau, and I think Quanterix can also comment on this, for tau we don't seem to see as much of an issue in terms of the stability of the tau protein over time in this assay or in the Simoa assay. Phospho-tau, we've actually started some new experiments to see whether or not if we add phosphatase inhibitors, if that might actually be able to boost the signal. We know that you don't need that for phospho-tau 181. It's measurable in CSF, and the assay is robust and reproducible, but it's entirely possible that for some of the other phosphorylated or post-translational modifications of tau, it might be important. So that's something that we're just started. With respect to ataxin-2, we don't know yet, but so far it doesn't seem to be impacted.